So we're continuing with cellular respiration today. And with cellular respiration, remember that the main idea of this, or the main idea of the process, is really to transfer energy into a usable form. And of course, that form is going to be our energy storing molecule, ATP. ATP is very easily used by most cellular processes, and this is something that happens with all cells. And ATP just happens to be that preferable molecule. Today we're going to focus on two steps, or two more stages of cell respiration. Oxidation of pyruvate, which is really stage 2, and the Krebs cycle, which is stage 3. But before we move into that, we want to think about how did we get there. And we first had to go through stage 1, which was glycolysis. Stage 1. I can't write it one. Stage 1 of glycolysis. So let's think about first, when we talk about the oxidation of pyruvate and the Krebs cycle, I do want you to keep in mind that this process, these things, we're actually going to be moving into the mitochondria. So take a minute to stop and try to answer these who, what, when, where, why questions on glycolysis. Try to refresh yourself and review what happened during glycolysis and what was most important. So when we're looking at glycolysis, the who performs glycolysis? And this literally was all cells. Prokaryotic and eukaryotic cells all perform glycolysis. And this should make sense because where did this occur? This occurred in the cytoplasm of the cell. And all cells have cytoplasm. Okay, they did not require a specific organelle for this to occur. Now, what did we end up making? We ended up making or producing our products. We ended up making 2 3-carbon molecules, or pyruvate. We made 2 ATP molecules, that was our net gain. And we also made 2 NADH molecules. One important thing to remember about NADH is this is our electron carrier. And the reason why this is so important, why this guy is so important, is because this is going to allow us to be later can be used to form ATP later on. So it is really important that we're slowly building up an accumulation of all these products, especially our carbon and our electron carriers, because we're going to use that all later to make more ATP. So when does this happen? Well, glycolysis again was that first stage, so whenever the cell needs energy, needs ATP. And then why does this happen? Well, what are we doing in glycolysis? We're really actually, and throughout this whole process, we're trying to extract energy from glucose. Okay, that's why we're doing respiration. We're slowly breaking down the molecule to get our energy. So looking quickly at a little overview of this, there's 3 fates to pyruvate, 3 things that can happen to pyruvate after glycolysis has been performed. You can go through 2 types of anaerobic respiration, which we talked about previously, lactic acid fermentation and alcoholic fermentation. And today we're really going to concentrate on this aerobic this aerobic oxidation. So we need oxygen for this process. One thing you want to focus in on is the word oxidation. This should remind you of those redox reactions. Remember that reduction means that we are gaining electrons, and oxidation means that we're losing electrons. And those always go hand in hand. The one really important thing here is looking at this acetyl-CoA molecule, okay? And we're going to talk about how this molecule can actually act as a form of regulation. Regulates when we actually have a lot of ATP, and we'll see this molecule later on, but I want to mention this now. When there's a lot of ATP available, this molecule, acetyl-CoA, will actually go through lipid synthesis. What does that mean? Well, if you think about it, when we eat lots and lots of carbohydrates and we don't need to turn it into ATP, what happens to all the carbs that we eat? Well, it turns into lipids or fats in our body. So this happens a lot with animals, or it's very easily seen in animals, that we're producing these lipids and storing this excess carbohydrate that way. Okay? So the next little section is just a quick review of the structure and function of the mitochondria. There's a couple little areas that I wanted to remind you of here. With mitochondria, important fact to remember is the endosymbiont theory. This theory said that mitochondria used to be a prokaryotic cell, meaning that it has its own DNA and enzymes. Remember that the mitochondria is found in all eukaryotic cells. And what we needed to keep in mind is with all eukaryotic cells, that does include our plants, animals, fungus, and our protists. They all have mitochondria, so they actually all can go through aerobic respiration. And with our mitochondria, we do need to pay attention to the intermembrane space and the matrix. We will mention this later on, so it is important that you know where those processes occur. So moving on, we have the oxidation and decarboxylation of pyruvate. Basically, this is step 2. Okay, so this is stage 2 of respiration. What's happening here is this is our pyruvate, and let's remember that this is what's produced at the end of glycolysis. Some things to notice in our title here. We have oxidation. What does oxidation mean? Hopefully you remember that this is the removal or the loss of our electrons. Decarboxylation, we're actually going to remove a carbon. And when we remove that carbon, we're going to find that we'll actually exhale that carbon as CO2, carbon dioxide. So what happens in the oxidation and decarboxylation of pyruvate? Well, you have your pyruvate molecule, and notice how these two carbons are in a different color. Remember that we are removing a carbon. So you actually have a transport protein. This is happening within the membrane of the mitochondria. Okay, in the membrane of the mitochondria, you have your pyruvate. It's a 3-carbon molecule. It goes through a transport protein, and actually that transport protein will, while it's moving through the membrane will remove a carbon. So you'll remove a carbon. And whenever this carbon is removed, we actually have to have oxygen present. So O2 must be present. We don't know why the oxygen is present, has to be present, but it just happens to be that it has to be there. If oxygen is not present, then this reaction will not continue. In the second step here, we have an NAD+. You're actually going to be removing some electrons from our pyruvate, okay, oxidation of the pyruvate. And when we oxidate the pyruvate, we actually form this product, the NADH again, okay? And again, this is from the removal of our electron. And then the final thing that happens here, so we remove the carbon, oxygen has to be present for that. We're going to remove electrons, the NAD will become reduced. It's gaining an electron and it becomes NADH. And then we have this final thing, coenzyme A. Coenzyme A is actually going to act as a method of delivery. These two carbons that are left over, notice how that carbon has been removed, the CO2 there has been removed. Once it's removed, these two carbons that are left are actually very unstable. Because it's unstable, we need this coenzyme A, notice SCOA, needs to bond with it to allow it to be carried on to the Krebs cycle. So these two carbons are unstable, and so we need this carrier to help move it to the Krebs cycle. Keep in mind that this process, remember how many pyruvates did we make? Well, we made 2 pyruvates at the end of glycolysis. So all this happens 2 times for each glucose molecule. Okay, all of that happens 2 times for each glucose molecule. So then we want to look very quickly at the Krebs cycle. I want to give you a little overview of what's happening with the Krebs cycle. We will talk about all these other steps at the top, but I do want to give us a little intro into the picture that's here. Really important things that we'll need to talk about is this oxaloacetate molecule and this citrate molecule. So at the end of the oxidation and decarboxylation of stage 2, after stage 2 occurs, we're going to move on to stage 3. And in stage 3, you'll have your acetyl-CoA. Remember, your CoA is helping to carry it. These 2 carbons will actually carry to this oxaloacetate molecule, this 4-carbon molecule. This was a 2-carbon molecule. And it will actually be used to form a citrate, which is a 6-carbon molecule. Then what we're going to see from here is that you're going to see a lot of carbon being removed. We're going to remove carbon, and we're also going to remove electrons each time we remove a carbon. And you'll see that happening once here, twice here, and actually a 3rd time. Oh, no, I'm sorry. Once here and twice here. And actually we'll see other areas in which we make more NADH, FADH2, that's the first time we're seeing that molecule. And down here we're also going to be making ATP. But we'll continue with this tomorrow.